Well, good morning to everybody. The topic of my lesson will be about the physiology of spermatogenesis. This slide shows the objective of the lesson that will be to know a little bit about the anatomy of the testis and the seminiferous tubule and the histology of the seminiferous tubule, the type and function of the cells that are involved in spermatogenesis, and how the testicular tissue is controlled by hormone. We will talk also about how spermatogenesis occurs within the seminiferous tubule. We also talk about the epididymis and the function of the epididymis that you will see is very important. The anatomy and the physiology of the main accessory glands present within the male genital tract that leads at the end to the composition of the complete ejaculate, of which we will talk. So this figure shows schematic organization of the male genital tract. The testis, which is surrounded by the epididymis, is located within the scrotum. From the epididymis, the vas deferens, that reach the seminal vesicles, which are located in a posterior region in the bladder, of the bladder. From the seminal vesicles depart the ejaculatory ducts that arrive within the urethra, from which the complete ejaculate is then emitted in the vagina, of course. From an histological point of view, this is how the testis and the seminiferous tubule is organized, also from an histological point of view. It is calculated that there are about 600 tubules for each testicle. And if we consider that one tubule on average is long about 60 centimeters, we can calculate about 360 meters of total length of tubules within the one testicle. The tubule represents from 60 to 80% of the total testicular volume. And from histological point of view, we can divide the histology of the testis in tubular compartment and interstitial compartment. Each tubule, as you can see here, is surrounded by the lamina propria, here in red, that contain mostly germ cells at various stages of development, and only one type of somatic cell, what is called Sertoli cell. Germ cells take strict contacts with Sertoli cells, and the ratio between Sertoli cells and germ cells is already programmed. In case of men is or human is only 4, as we will say also later. And the aim of Sertoli cell is that to support and to they are also known as the nutritional cells and sustenticular cells, and are cells that support spermatogenesis, support the development of germ cells until they become spermatozoa. Sertoli cells also contribute to the blood testicular barrier, so that blood should never get into contact with germ cells in a normal testicular testis. The lamina propria is also important for the phase of spermiation, that is the release of sperm cells from the epithelium, because they promote peristatic contraction to allow sperm cells to leave the epithelium. And in the interstitial compartment, are present Leydig cells that are endocrine cells that produce testosterone. Testosterone that then will be released into the blood, but also will reach the tubular compartment because the hormone also support spermatogenesis. And in particular, Leydig cells respond to testosterone and the other hormones that support spermatogenesis. But in the interstitial compartment are also present lymphocytes and macrophage that produce other substances that may be important for spermatogenesis, like cytokines, and also connective tissue, lymphatic, and of course blood vessels. In human, spermatogenesis lasts from 70 to 90 days, and this is very important to know the length of complete spermatogenesis. For instance, when a man assumes drugs that may temporarily produce a damage to spermatogenesis, it's important to know how long does it last, because we must advise this man before that he undergoes a semen analysis to wait that at least a complete cycle is finished. And so, for instance, a man that takes antibiotics should not undergo semen analysis, or, for instance, also an ART cycle before 90 days from the end of assuming antibiotics. So how spermatogenesis occurs? Spermatogenesis will start from primordial germ cells and spermatogonia. We will see later on that there are 3 different types of spermatogonia. These cells undergo a mitotic division, producing a cell identical to itself, but also primary spermatocytes, which are still diploid and undergo the first meiotic division, producing secondary spermatocyte, which undergo the second meiotic division, producing spermatids. Spermatids are haploid cells. They cannot divide anymore, and they cannot even produce proteins. So the protein synthesis is stopped in spermatids. And spermatids are the cells that undergo the last phase of spermatogenesis, which is called spermiogenesis, that leads from a round cell, like spermatids are, to a sperm cell, which is a flagellar cell that then will become able to move. This slide shows the complete spermatogenesis and the number of stages that have been recently identified in men. We know now that the stage are 12 and not 6 as previously believed, according to other studies. And this is thanking to the group of Stefanini and Elena Vicini in Rome. This study is actually very important because it's the one that finally defined the number of stages also in human, and demonstrating that reflected those occurring in many other mammalian species, such as, for instance, the mouse. Here are also shown the 3 different types of spermatogonia that are present in human tubule, and that are the AD, the AP, and V. AD stands for dark, are normally quiescent cells and are the true stem cells present in the testes. They can initiate mitosis only following particular stimuli, such as, for instance, irradiation, and there are many studies demonstrate this. AP or apale cells are also quiescent, but they become mitotic following stimuli, hormonal stimuli. And the B cell is the product, which are B cells are those from which originate the spermatocytes and from which spermatogenesis progresses. This is a schematic illustration of the complex process of spermiogenesis, which leads, as I said before, from a round cell to a flagellar cells. During this process, during the process of spermiogenesis, some structures that are important for the sperm cells and for fertilization of the oocytes are formed. The first one is the acrosome. The acrosome originates from the Golgi apparatus, the Golgi vesicles that coalesce together to form this structure that is called acrosome, and that stands like a cap over the nucleus in the final sperm cell. The acrosome contains enzymes that are very important to cross the investments that surround the oocyte. The other important structure that form during spermiogenesis is the tail. The tails form starting from centrioles that act as a template for the assembly of cytoskeleton elements that form the tail. During spermiogenesis also occurs the first part of the nuclear condensation or chromatin condensation that consists in histone to protamine transition. At the end of the process, basically all the histones that are present in the chromatin are substituted by protamins. Protamins are basic proteins present in the chromatin. And at the end, only 5% of histone remains into the chromatin, in the mature chromatin of a spermatozoon. So the final product of spermatogenesis is the spermatozoon, which has been defined by somebody as the most highly specialized cell type ever described, and is certainly one of the most specialized cells present in human beings. Here also a schematic illustration of how of the composition of human spermatozoa. Basically, we have an head, which is formed by an acrosome and the nucleus. The tail, which is composed by the midpiece, inside which there are the mitochondria, which are very important because they produce the ATP necessary for the movement. But ATP is not coming only from mitochondria, it's also coming from the glycolytic process that occurs along the flagellum. The principal piece and the end piece. Along all the flagellum, there is the axonema, which is formed by 9+2 doublets with DNA in arms and tubular proteins, of course. The axonema is very similar to the one that is present, for instance, in bacteria flagella. So how is regulated testicular function from an hormonal point of view? This slide shows the schematic representation of the hypothalamus-hypophysis testicular axis. The regulation initiating the hypothalamus, which produce gonadotropin releasing hormone, GnRH, which stimulates the hypophysis, the anterior hypophysis, pituitary gland, that to produce the two gonadotropin, LH and FSH. LH is the luteinizing hormone, FSH is the follicular stimulating hormone. FSH acts at the level of Sertoli cells, which possess a receptor for this hormone, to stimulate and to promote the spermatogenesis process. LH, on the contrary, acts at the level of Leydig cell to stimulate production of testosterone. The axis is regulated by a negative feedback, which is set by testosterone. Testosterone exerts a negative feedback on the pituitary gland to limit the production of LH. Whereas FSH is negatively regulated by a feedback exerted by estradiol, which is also produced at the testicular level, and by inhibin, which is an hormone produced by Sertoli cells, which give information to the pituitary gland to stop or to limit the production of FSH, because spermatogenesis correctly proceeding and going on. But spermatogenesis is not only regulated in this way. We know that there are many different genes that control spermatogenesis. It is estimated that for spermatogenesis it is required the expression of about 2,000 genes and 2,000 proteins product of these genes. And these proteins may act both in a paracrine and autocrine way. And many of these genes have been knocked out in mouse, and demonstrating that when they are knocked out, spermatogenesis is either less efficient or is completely stopped. So demonstrating that all these genes are very important for spermatogenesis. And at the end of the story, human spermatogenesis is not so efficient, and for several reasons. First of all, because the number of Sertoli cells is lower compared to other species. The ratio Sertoli germ cells is only 4 in human, as I said before, whereas, for instance, it's 10 in the rat. The number of produced spermatozoa, which is expressed as million per gram of testis in 24 hours, is from 3 to 7 in humans, whereas, for instance, is from 10 to 24 in the rat, which is actually much greater. Not all germ cells arrive at the final product, and it is estimated that is about 50%, and this is because the apoptotic program in the testis is very efficient, not only because it's important to maintain the ratio Sertoli germ cell, but also to eliminate other cells that are not completely normal germ cells that are defective in the spermatogenesis production, in the spermatogenesis. And finally, we know that most human spermatozoa are morphologically anomalous, are not really typical, like it happens in other species. But spermatozoa, when they are released from the testes, are not completely mature. They must then undergo 2 post-testicular maturative events in order to be able to fertilize an oocyte. The first maturative event occurs in the epididymis and is known as epididymal maturation. And the second event occurs within the female genital tract and is known as capacitation, during which spermatozoa completely achieve the ability to fertilize an oocyte. So what happened in the epididymis and what is the epididymis? The epididymis, which is here represented in red, is a tubular structure that surrounds the testes and that take contacts with testicular tubes to the efferent ducts. It is composed by an initial segment, here the caput, the corpus, the cauda. The cauda is in contact and continues in the different duct here. Within the epididymis, we recognize different type of histological cells. See here some of them. The principal cells, which are present in the entire epididymal duct, are involved in fluid reabsorption and secretion of many different proteins and epididymosomes that are involved in this maturation process. The apical cells that are mostly present in the initial segment have and are shown to have endocytic activity. Narrow cells, which are also present in the initial segment and are involved in secretion of hydrogenium to acidify the pH in the lumen. Clear cells here that are present in the caput, in the corpus, and in the cauda, and are also involved in the luminal pH regulation. Basal cells here in gray and black are present along all the tubule and are involved in the protection from in secreting substance that protect from oxidative stress and other possible environmental factors that they may encounter in the male genital tract and even in the female genital tract. Halo cells that are present in the epididymal epithelium are cells like T helper, T cytotoxis, and are involved in protecting from the generation of anti-sperma antibodies. Each of the epididymis parts have a specific has a specific function. The caput is involved in fluid reabsorption and in the formation of disulfide bridge within cysteines present in protamines. As I said before, protamines are basic proteins that contain many cysteines, and what happens in the epididymis, both in the caput and in the corpus, is the formation of a bridge disulfide bridge within the cysteines. And this is very important to complete chromatin compaction and so maturation of the chromatin to maintain DNA integrity, as I was saying before. So this occurs both in the caput and in the corpus of the epididymis. In the corpus also many proteins are synthesized and these proteins are involved in the acquisition of progressive motility together with substance like as calcium and bicarbonate, and also defense proteins that are involved in defense response of the sperm. In the cauda probably has a role as sperm storage, is also involved in the last phase of sperm chromatin compaction, has a defense responsibility, and it is believed that is probably involved in a sperm quality control, that is elimination of anomalous spermatozoa, although we are not sure that in human epididymis such process is so efficient like in other animal species. So as I said before, the epididymis is producing many proteins, and these proteins are secreted within vesicles that are called epididymosomes. The epididymosomes have many roles. They are involved, you see, in all the post-testicular sperm maturation. They are involved in epigenetic modifications within the nucleus, in transfer of crucial fertility and modulating protein to the sperm surface, and even they alter RNA present in sperm during epididymal transfer. In particular, epididymosomes transfer protein that may adhere to the surface of the sperm. These proteins are called decapacitation factors and are then lately removed during the process of capacitation to allow exposition of proteins that are involved in sperm oocyte recognition. So this is a very important process to protect this protein during the transit in the male genital tract and not then remove in the female genital tract. So let's go to the composition of the ejaculate. Of course, in the ejaculate, there are cells coming from the testis, not only spermatozoa. We will see later on that also immature germ cells and apoptotic bodies are coming from the testis and may be present in the ejaculate. Composition of seminal plasma. Seminal plasma is composed by many substances, and from 65 to 80% of seminal plasma is produced by seminal vesicles. About 20–30% is coming from the prostate, from prostatic fluid, and only 1% from bulbo-urethral glands that are in a position after the prostate. Staminal vesicle produce substance they have a role as nutrient for spermatozoa, such as fructose, citrate, ascorbic acids, but they also produce prostaglandin and semenogelins that are clotting proteins. So they are involved in the formation of the clots that is how the ejaculate is at the beginning. Prostate produce clotting enzyme also, but also protease that are involved in what is happening after ejaculation, that is the liquefaction of the ejaculate. They also produce nutrients like citrate, but also buffering substance such as bicarbonate and zinc. Zinc is very important because contribute to maintain chromatin compaction. The bulbo-urethral glands produce a lubricant, a mucus, and buffery substance that neutralize the acidic pH in the vagina. In the ejaculate are present also somatic cells like leukocytes, macrophage, epithelial cells, but there must be also present, for instance, erythrocyte. These cells are coming from the ducts, the deferent and the urethra, when, for instance, inflammations are present there. Epithelial cells are cells that are also found in the urine, and they are also coming mostly from the urethra. The pH of the ejaculate should be maintained between 7.2 to 7.8 or 8 at maximum. The liquefaction duration is usually from 30 to 60 minutes at maximum. If an ejaculate does not liquefy within 60 minutes, it means that protease produced by prostate are in a minimal quantity and do not support complete liquefaction of the ejaculate. So what type of cells we can see in the ejaculate besides, of course, spermatozoa? We can see germ cells, and actually in the ejaculate, all the type of germ cells during that are formed during spermatogenesis can be found, starting from spermatogonia to a spermatide, and different type of spermatide. So many type of germ cells can be present in the ejaculate. As I said, are also present apoptotic bodies here that are easily recognized after staining in a Megra Grunwald, like here, because they don't have a nucleus. What are apoptotic bodies? Apoptotic bodies are the product of an apoptotic process. As I said before, in the testis, there is a very efficient apoptotic pathway, apoptotic program, which, however, is not completed. And this is the reason why both dead germ cells and apoptotic bodies are present in the ejaculate, because these elements have not been endocytosis by the Sertoli cells or macrophages, and so they are present in the ejaculate. And somebody actually call the apoptosis that occur in the testis abortive, because it does not complete like it happens in other tissues. As I said, in the ejaculate are they might be present leukocytes, and leukocytes are easily recognized after staining, like here in Meigrum, but also other types of staining, and usually indicate the presence of an inflammatory problem within the testis. And here also are shown epithelial cells that are usually coming from urethra or deferent. Here is how an ejaculate appears under the microscope. This is a 400 per microscope. As you can see, we see cells, but we see spermatozoa moving. This is a normal ejaculate. This is also a normal ejaculate. Actually, here, as you can see, there are many, many moving cells, and they move also progressively and very rapidly. Here, there are cells that move less rapidly. And this is another type of ejaculate that, as you can see, here there are many cells that are immotile. So anyway, these are three, I would say, quite normal ejaculate, because of course there can be very abnormal ejaculate in men. So at the end of this story, which are the reference values when we perform an analysis of the ejaculate, a semen analysis? At present, we are using the reference centile table, which is indicated in the last version of the World Health Organization semen manual for human semen that has been published in 2021. The manual is using the distribution of semen examination results published in 2021, and which collated together the results of a population of fertile men coming from different countries and different continents, as you can see here. These data have been collated together, giving rise to this table, which report the data for more than 3,500 fertile men. What is important to know is that these men are fertile, securely fertile, and obtained a natural conceivement within 12 months from starting of the desire of having a conceive. And this table report centile from the 2nd to the 97th with the values for each of these parameters, that is the semen volume, the number, sperm concentration, total number, the total motility, progressive motility, non-progressive motility, that is immotile sperm, the in situ motile sperm, immotile spermatozoa, viability, and normal forms. That is the morphological evaluation. Until the previous laboratory manual from World Health Organization that was published in 2010, it was decided to utilize the 5th percentile of a similar table that was published in 2010 together with the manual. The difference here is that the number of fertile men in this paper has increased from 2,000 to almost 3,600. But I would say that the table is very similar, confirming that fertile men showing a time to pregnancy below 12 months have similar semen analysis parameters. And so this was very important for World Health Organization, demonstrating that also the values that were shown in the previous manual were very similar. But what is important to know is that in the 2021 manual, it has been decided to avoid to use a reference value. But what they are asking now in this manual is to use this table where are reported the centiles. And so this is how a report of a semen analysis should be. This is ours. I'm sorry, this is in Italian, but what I want to show here is that there are no reference value, with a few exceptions, like the abstinence period, which should be maintained between 2 and 7 days, sexual abstinence period. The number of leukocytes, which should be maintained below 1 million per ml of the ejaculate, but mostly there are no reference value. What we report here is the table that we have seen before, reporting the different values for the centiles, the percentile between 50% and 95%. And this is followed by an important note. The note says that these values are not to be intended as a reference value, and thus they do not represent a limit between fertile and infertile men. This is because this is a distribution of semen parameters of a fertile population that obtained a pregnancy within 12 months. And this is very important. And the reason why it has been decided not to use these values as reference values is very simple. It is because, as far as we know, semen analysis cannot take and cannot say us which are all the molecules that are useful and that are necessary in order for sperm oocyte interaction and sperm fertilization of the oocytes. There are many molecules. We don't know from the semen analysis about the integrity of the DNA that is transmitted to the oocytes, and there are many other problems that may occur later on in the female genital tract that not necessarily support the capacitation of spermatozoa. So it appears quite clear that within the table, these tables, these men are all fertile, either that they have values in the 5th percentile or the 95th percentile, but we know for sure that there can be men that have values in the 95th percentile and can be infertile, as well as that there can be men that have values within the 5th percentile and can be fertile. So it is important to know this fact. And So this is the end of this presentation. Of course, if you have questions and comments, I will be glad to answer. Please write to me at this address. And so I thank you for listening to this lesson.